Hello, everybody. You might have noticed that Karthik announced Daniel as our Lenary speaker. This is actually no mistake, because I've heard from the organizers that Daniel's predictions are so accurate that the P could be ignored. But seriously, Daniel is an incredible scholar. He has produced excellent research, very influential research on a diverse array of topics: evolution of cooperation, decision-making under uncertainty, and the effects of early life stress on developmental outcomes. He's done both theoretical and empirical work that's highly integrative as well. So he writes, for instance, articles for behavioral ecology, philosophical transactions of the Royal Society, where he really brings together different approaches that we all take to our field. So I think that's wonderful. And one thing that maybe not many people here will know is that Daniel is also highly creative. So he writes plays, for instance, in addition to science. And he told me just this week about a study that he's planning that's also highly creative and integrative, because he wants to do a study in which participants are endowed with $10, which they can then transfer some fraction of that to a bank teller called Linda, who is holding in her left hand a can of hot sauce, and with her right hand can switch a train go into 5 different people while a gorilla is jumping in the background. So he's highly creative. What I definitely should not forget to mention is that he is a major contributor to our community at large. So he's an editor of the flagship journal in our field. He's the president of the European equivalent of this society. So he's really making major contributions of the highest quality to our field. On a more personal note, I would say that Daniel is also a wonderful collaborator. He always is very constructive, very precise. He's a very clear thinker, and he also writes very clearly and beautifully. So that's much appreciated. It appears that archaeologists recently discovered a new cave where they found a quote. It was in ancient Greece. There was a quote that stated, "Enter the Nettle and the dust will settle." Ladies and gentlemen, Daniel Nettle.
Thank you very much, Willem. I don't quite know how to follow that, and possibly I shouldn't, actually. That might be the best way to do it. But thank you for your very kind words. My talk today is about early life adversity. So the topic I've really been concerned with for the last 5 years or so, above all else, is how does what happens to us early in life affect our outcomes later on? So if we consider children who have adverse early experience, for example, these kids from a Victorian workhouse, the chances are that their lives will be very different from their peers who don't go through this stressful experience. For example, they may have worse health as adults. There's lots of evidence of that. And their behavioral phenotype may be different too, in lots of ways that we'll talk about. So what we really want to understand is at both a proximate and an ultimate level, what's going on here? Why is there this plasticity such that what happens early in life can end up being so influential? I think it's a good idea at this point in the talk to try and tell you what my big idea is, in as much as it is a big idea. Because if we get that out of the way early, then, if we have to stop now, at least you got that in. And I'm going to attempt to explain my big idea for thinking about these early life effects in terms of novels about pre-20th century Paris, which has seemed the natural way of doing it, obviously. So the first component of my kind of approach to these things is that an adverse period of ontogeny damages the adult soma, okay? So you're trying to build your tissues, you're trying to grow, you're trying to develop, and if during that critical period, which you only get to do once, the inputs are inadequate, the parental investment is inadequate, you face a high disease burden, or any of various other bad things happen to you, you're simply able to build those tissues less well. So you end up with more kind of damage by the time you have an adult body. This kind of part of the diagram is represented there, that early life adversity damages the adult soma. And so my novel reference here is Dickens' A Tale of Two Cities. In book 1 of that novel, our heroes are turning up in Paris on the overnight stagecoach, and they pull into this kind of poor neighborhood. And they start to see people kind of come out of the gloom, and the people look terrible. They look, absolutely sort of used up by the adverse conditions they're living in. And what Dickens says is, the mill that had worked them down was the mill that grinds young people old. So he's doing something very interesting here, which is making a link between adversity and aging. And what he's actually saying is, is that if you have this adverse ontogeny, the processes of aging are sort of speeded up, that this mill grinds you old. And given that the biological definition of aging is the accumulation of basically random molecular damage that leads to a decline in biological performance in the soma, this is actually quite a prescient kind of idea that this adversity is causing these people to age. Hans Selye, the father of modern stress physiology, said something very similar. Every stress leaves an indelible scar, and the organism pays for its survival after a stressful situation by becoming a little older. So the basic idea here is early life adversity may simply leave you with this kind of prematurely aged soma. So that's one part of the diagram. The second part here is the idea that there may be a link between the kind of somatic state you have and the behavioral phenotype you adopt. And the reference here is L'Assommoir by Émile Zola, which is a novel, again, about proletarians in Paris who get involved in various kind of situations, who've had these terribly rough early lives. And something that's very striking about them is they have this incredibly impulsive phenotype. So they get some resources. They always go for the most immediate thing to do with them, which is usually alcohol. They get into all kinds of debts and scrapes and making various kind of short-termist decisions which keep them in the game of living in this community, but ultimately, it all ends in tears. So the idea here is that if you've had an adverse early life and your soma is in a bad way, so that you're likely to be less competitive, you're likely to be less vigorous, you're likely to be alive less long because it's going to start falling apart sooner, it may actually be adaptive to kind of calibrate your behavioral phenotype to that. What kinds of resources are you going to compete for? What kinds of behavioral decisions are you going to make? Well, you ought to calibrate those to the state that you've got. What kinds of things are you likely to be successful at? What kinds of investments are likely to pay off over the time scale at which you'll be in a position to enjoy them? So, whereas this part of the diagram is all about pathology, this part is all about adaptation, conditional adaptation to the kind of body that you have. And that's the basic approach I take to these kinds of problems. I'll just say very briefly, this is a rather different approach to the kind of theoretical model that's quite common in evolutionary psychology, you see, where people say, well, the early life experience you have is giving you information about the environment you're likely to inhabit when you're older. I call those weather forecast models. So if your parents are behaving in a certain way, it tells you something about the environment that you're likely to inhabit later on. That's a rather different claim. And in particular, I think that claim is kind of problematic in long-lived organisms such as ourselves, because it buys into very strong assumptions that over evolutionary time, the environments of ontogeny have been highly predictive of the environments of adulthood. And I think in fluctuating environments, that's a potentially problematic assumption. This kind of view of things doesn't buy into that, because we're not saying that your early life conditions are predictive of the external environment that you'll experience in adulthood. What we're saying is, what happens to you is going to leave its scars in your body, and you're going to have to calibrate your behavior to the kind of body that you've got, which is a rather different claim. I think there are distinctive predictions between the weather forecast view and this more somatic view, but rather than spend time on them now, I just point you to some papers that Willem and Ian Rickard and I have written on that topic. But I suppose a distinctive prediction of my view is that to the extent that there are relationships between adult behaviors and early life adversity, they should be mediated by measures of adult somatic damage. Right? So this should be a kind of common final pathway linking all kinds of adversity in early life to later behavioral phenotype. And we'll see some tests of that in this talk. Okay, so the other unusual thing about my research program is I do it comparatively in two species, both illustrated on this slide. These are two long-lived social vertebrates that both thrive in the north of England, both like fish and chips. Humans, of course, and the European starling. And this is a really exciting thing to do because these two species offer us very different possibilities for what we're able to do. In the human, of course, we've got a wealth of very detailed epidemiological longitudinal data. We've got wonderful cohort studies where people have measured all kinds of things in the same cohort over many years. And of course, in humans, you can ask them to tell you about their childhood and the conditions of their life and what kind of decisions they make and so on. So all that's very convenient. But the human work has a tremendous limitation, which is it's inevitably correlational. So if we find in humans associations between some early life situation, let's say family composition in childhood, and some later outcome, let's say age at menarche, we simply can't conclude strongly that there's a causal relationship there. Both could be the product of some unmeasured third factor, which could be genetics, for example. So you might have a genetic correlation between the propensity to have certain types of family structures and the speed of your pubertal maturation, or they might be products of some third factor like economic poverty, for example, without there being any important causal link between the two. You can't do experiments with humans in this area. What you'd ideally like to do is take pairs of twins on the day after their birth and separate them to have them grow up in different families or different socioeconomic conditions, and obviously that's ethically and practically impossible. But it's not impossible in an animal model, and that's exactly what we can do with the European starling. And so really the thrust of this work I'm trying to do is saying, what can we do experimentally in an animal model that might lead us to think about these processes in humans that we can only study in a correlational way? Now, you might find it a bit odd that I use a bird and not a mammalian model, a rat or a mouse or a monkey or something. Actually, birds have some tremendous advantages for this kind of work. So there we go. So one of the points to make is that passerine birds live a long time. So a European starling can live for 20 years. It's a 75-gram animal. And it's got a slow life history, right? So it only reproduces once a year. So in a way, if you're interested in how things that happen very early on can have really rather delayed effects in a long-lived animal that's likely to experience all kinds of different environments over its long life, then actually your bird model is going to probably do better than a rodent model, for example. But just as they're long-lived, there are some other peculiarities of their life history we can exploit, that the developmental period is enormously compressed. So birds have this wonderful adaptation, flight, which means that they enjoy a relatively low rate of extrinsic mortality. They can evolve very slow life histories for their size. But the period before they can fly, they're enormously vulnerable to predation. So selection has really curtailed the developmental period to an incredibly small proportion of the total lifespan. This is an animal that can live for 20 years, but it attains its adult weight in 10 days, which is remarkable. And it's flying around independently in 21. So at the same time we have a long-lived organism, we actually have a very short window where the important developmental action is happening. And that's actually very important or very useful experimentally because we can, in principle, create experimental manipulations that last for pretty much the whole period of its linear growth, of its main kind of developmental period. They have some other advantages, which again we can exploit. The first thing to say is that starling chicks and their parents don't recognize each other for about the first 14 days of life. So we can very readily do cross-fostering, move them from one nest to another, or we can take them into captivity and have them imprint on us, and then we can be the parents. And then, of course, we can very tightly control exactly what kind of investment goes in. And at the same token, of course, in any mammal, parental investment is primarily delivered through lactation. So you've always got this problem if you want to do, for example, a manipulation of nutritional inputs. You can't do it directly. You have to do something to mum and hope that that kind of flows through to the pups, which it may or may not do. She may buffer it. Whereas in the starlings, everything is being delivered manually into their mouths, and so we can see exactly what's going in, and we can experimentally manipulate it. Okay. So throughout this talk, I'm just going to present a few correlational studies in humans and some related experiments in starlings, just showing you how we can go back and forth between these two systems to study the kind of processes that I've described. And in this first empirical part, I'm really talking about the left-hand side of my diagram, that mapping from, here it is, from early life adversity to adult somatic state, with the particular idea that what adversity does is accelerate the process of biological aging. So I've studied this in humans. I've studied it in particular in a cohort of British women called the National Child Development Study. They were all born in 1958, and they've been followed up through life. And working with David Cole and Tom Dickens, we developed a really super simple index of early life adversity, which was characterized really by shortfalls in parental investment or relative lack of parental investment. And these were not big things. These are relatively subtle things. Not being breastfed was one of them. Dad not being around was another. The third was being separated from mum for more than a month at any point during the first 7 years of your life. And the fourth was the family moving house a lot. So these are kind of small psychosocial stressors. And we just made the simplest possible index, which is to add up how many of these bad things happen to you from 0 to 4. And in this paper, I went back to look at the health of those people when they were 42, because we had the data on the early life stuff from the period 1958 to 1965. And what you see is that the more of these childhood adversities you have, the poorer your self-rated health is. All those years later, when you're 42, and the higher the rate in your blood of C-reactive protein, which is a sort of general marker of non-specific inflammation. Right? So inflammation is very much tied up in all kinds of poor health, and more of it is generally considered a marker of kind of poorer condition. And as you see, you get this effect that the more of these adversities you have, it's an increasing inflammation, decreasing health. And this was robust to control for obvious things like socioeconomic position. Now, the interesting thing about this is that these are things that change as you get older. So basically, and you can see this longitudinally in that very same data set, basically as you get older, you lose about 0.25 on this scale with every decade that you get older, and this goes up by about half for every decade that you get older. So really you can think of these groups who had the 3 and 4 adversities, they're a decade or 15 years older than the people who had the 0 and the 1. But of course that's correlational evidence, and what I really want to talk to you about today is whether we can experimentally investigate some of these processes. So here's the system with which we do it. We're going to do experiments in the starling that I'll now explain, but first we need a marker of some health-related outcome that we can consider a kind of integrative measure of biological aging. And the measure we use is the length of telomeres. So telomeres, you may be aware, these are the repetitive non-coding sequences found at the ends of all eukaryotic chromosomes. They shorten as we age. They're particularly vulnerable to oxidative damage. And as we get older, our telomeres generally get shorter. Obviously, in every cell, you have multiple telomeres, and they can all be different lengths from one another. And in every tissue, you have multiple cells. But there's been a lot of interest in the last 20 years in the idea that you can take a kind of average length of the telomeres in a tissue as a measure of the kind of oxidative damage that increases as we get old. I won't go so far as to say it's a measure of biological age, as some have, but it's some sort of measure of the accumulation of damage as we get older. Now, there's variation between tissues in the length of telomeres, but that's absolutely dwarfed by large variation between individuals in the average length of their telomeres, and that's what we're going to use as a biomarker. So we're going to take blood from our starlings and measure telomere length in ways that I'll explain, and that will be our marker of kind of the aging that's been caused by your ontogeny. So we have a colony of around 100 nest boxes. These are wild animals, though we do sometimes take them into captivity, as you'll see. And because they're all in these nest boxes on farms, we're able to do cross-fostering experiments and other kinds of manipulations during the period when the chicks are small. So what I'd first like to tell you about is an experiment we did in 2013, in which we were trying to get at whether stressful adversities very early in life will accelerate the rate of telomere attrition. And this was the design. So from donor nests in our colony, we take 4 siblings. So this is important. This is central to all of our experiments as we're doing comparisons across sibling pairs who've been given different experimental treatments. So we take 4 siblings on day 3 after hatching. So the birds are about 15 grams. And then we cross-foster them to 2 different nests. The 2 siblings that go into the advantaged ADV condition go into a nest where all of the other chicks are about 5 grams lighter than they are. Whereas the other 2 siblings who get the raw deal and go into the disadvantaged condition go into a nest where all the other chicks are about 5 grams heavier than they are. These are not to scale, by the way. But it's a small difference. Now, why do we do this? We know a lot about the basic behavioral ecology of these birds. Parents feed their biggest chicks first, okay? So parents take a sort of insurance-based approach that, that their biggest, most vigorous chicks get fed first, and the smaller guys have to kind of fight for the scraps. And so that, if the food supply proves inadequate, the smaller guys die, but you get a successful heir, as it were. So that's the kind of parental strategy. The chick's counter strategy is that if you find yourself small and hungry, you beg like crazy, right? So it's well understood that the smallest chicks in the within-brood hierarchy have to beg more to extort investment from their parents. So, these guys in the disadvantaged condition are going to get a rougher time because they're going to be the smallest chicks in their nest. To get their parents to invest in them, they're going to have to work a lot harder. in terms of begging. And we leave them in this cross-fostered manipulation to day 12. So it's just 9 days of life. So that's about halfway through the period when they're in the nest. After that, these birds were taken into captivity. So what do we see in terms of the growth curves? Well, the red and the gray lines here are the growth curves of those disadvantaged and advantaged groups. In fact, as you see, during the period of the experimental manipulation, the cross-fostering happened here, and we took them into captivity here. There's absolutely no difference in their rate of growth. So they were getting the same amount to eat. I mean, this is the result. It wasn't something we necessarily expected. We thought perhaps the disadvantaged birds would struggle to get so much to eat. But actually, that doesn't seem to have been the case. Their growth curves tracked each other perfectly. But what you'll see from the dotted lines here, this is the mean weight of the competitors in the nest they've been fostered into. So the disadvantaged birds remain smaller than the ones they're competing with. The advantaged birds remain larger. In fact, growth is remarkably linear in this experiment, as you can see. So they got just as much to eat, but we hypothesized that they must have had to work much harder to get it. They must have had to do a lot more begging, and that's stressful. What about their telomeres? Well, we measured telomeres at day 3 from blood, and again at day 12 when the manipulation ended. And what you see here in this panel, so this is our measure of telomere length, is that it's significantly longer in the advantaged than the disadvantaged group. Now, I say our measure of telomere length. It's a slightly grandiose way of putting it. We use a very simple qPCR-based assay, which is basically conceptually equivalent to putting all the DNA in a bucket and saying what fraction of this seems to be the telomeric sequence. So we don't actually measure the length of any individual telomere. You just sort of stick it in the bucket and see what you can amplify. But this gives you a measure called the TS ratio, which is usually taken as a sort of a proxy for telomere length. And as you see, there's actually quite a significant difference. After just 9 days of this treatment, which is actually remarkable because it's really a very short time for something genomic to change. Now, you would hope that's not because their telomeres were already shorter at day 3, and indeed they weren't. In fact, what we find a relationship that's often been found in the telomere literature, that this is your telomere length at day 3 here, that the longer telomeres you had at the beginning, the faster they shorten. So this is the amount you lose. And this is how long they were initially. And as you see, there's these negative relationships. The longer they were, the faster they shorten. This is actually a mechanistically well-understood relationship. It's to do with there being more oxidative targets in longer telomeres. But what you see is that the advantaged and disadvantaged birds are on a different grade there. So for their telomere length, they're losing more, which is actually kind of a remarkable demonstration, because I think no one had dreamed that telomeres would be changed by stress in such a short space of time. Okay. Now, what that experiment suggested was that it wasn't how much you got to eat, it's the kind of psychosocial stress involved in getting it, because the growth curves, as you saw, were the same. But actually, the nest box was a black box to us in that experiment. We put the cross-fostered birds in on day 3, and we got them out on day 12, but we didn't see what the dynamics actually going on in there were. So we decided to do a more ambitious experiment to really try and tease apart, is it the nutritional thing, what you're getting to eat, or is it the psychosocial stress thing, how hard you have to work to get it? So we decided that we'd play the parents, and in an experiment we've just recently completed, we hand-reared birds, experimentally assigning them to get different schedules of begging and of food across the ontogenetic period. So this is a classic kind of 2x2 factorial design. What we wanted to do is to say, do you get a lot to eat or little? We call that our plenty and lean treatments. And is it easy to get what you get, or is it hard, in the sense you have to beg a lot for it? And we call that's our 2 work treatments. It's easy to get it, or it's hard to get it. We took the birds from the wild on day 5, which is the earliest we dared, really. I think it's difficult to rear starling chicks from much earlier than that. And could we have the video there going, please, if you can? This is where they grew up. So again, we take quartets of siblings from a nest, and we assign one each to each of these 4 conditions. So they're in these nest buckets. These are the 4 nests. And this is a slowed-down video to give you some sense of what goes on. We play the parent. We come and visit the nest. As you see, that causes the chicks to beg. They imprint on us, obviously. You'll see in a minute the feeding going on. So they've seen Melissa arriving, I think, and they're begging. We're feeding them here with an Eppendorf repeating pipette, so we know exactly how much is going in. Right? So we can measure in every feed exactly how much you're getting. And we can also manipulate how much you have to beg. So you can see the basic setup there. So there's 4 conditions in this experiment. In the plenty easy condition, which is plenty of food to get and it's easy to get it, we visit that nest 10 times a day. And on every feed, every visit, we feed each chick to satiety, until they just don't want any more. So they don't have to do too much begging and they get as much food as they could possibly want 10 times a day. And then in the lean easy bucket, there's a sibling of someone in the plenty easy bucket. And they get 10 visits a day, but on each of these visits, we feed 70% of whatever the sibling in the other bucket got. Okay? So we know on that, we can actually titrate that on a feed-by-feed basis because we've just written down what the plenty easy sibling got, and we just give 70% of that for this guy. So he doesn't get so much, but it's still relatively easy to get it. The third condition is the plenty hard condition. Here we do 20 visits a day. The 10 visits are identical to the plenty easy condition, so they're getting fed just as much and just as often. But in addition, between times, we make 10 other visits where we stimulate them to beg using the pipette for 2 minutes, but without actually delivering anything. So they get just as much food, but they have to do all this additional begging, which they obviously do when they see us. And then finally, the sort of toughest gig of all is the lean hard condition. They get the 20 visits a day, but they only get 70% of what these guys get, and they also get to do the begging as well. So that's the kind of double whammy. If we consider the growth curves of these guys, so we start here on day 5. Very rapidly, as you'd imagine, the 2 plenty conditions and the 2 lean conditions diverge. The lean conditions don't put on weight so quickly, of course. That would make sense. They're not getting so much to eat. They do eventually all get there in the end, right? So basically, as adults, these birds are all the same size. Unfortunately for us, in an ideal world, the lean easy and the lean hard would have had exactly the same growth curves as each other. That didn't happen. In fact, the lean hards fell behind. I think that tells you something about the cost of begging. There's an energetic cost to the fact they're having to do all this begging. It's a flaw in our experimental design, because if we really want to say we've cleanly separated the begging from the nutritional, the energy balance issue, then ideally, you'd want your lean easy and lean hards to have the same growth curves, but it didn't work out that way, unfortunately. So that's a slight kind of flaw in the cleanness of the design. So let's look at telomere attrition. So we're measuring telomeres here on day 4 and then again on day 15. I just should say something about this attrition measure. So this is a rescaled measure where 0 is the mean. For various reasons, we have to correct for a number of things, and so we end up with a scaled measure with a mean of 0. So 0 on this measure does not mean your telomeres didn't change. It means your telomeres shortened by the average amount in that sample. And so if you have a more positive value, they shorten by less than the average amount in that sample, and a negative value, they shorten by more. I hope that makes sense. So what do we see? Well, we actually see some very nice effects. So the plenty easy, these guys didn't have to work and got plenty to eat. They had the least telomere shortening of all. And the lean hards, they didn't get much to eat and they had to work for what they got. They have the most of all. And then intermediate are the 2 groups who had one or the other. So what that actually suggests is that there are 2 additive things going on here. It's bad if you don't get enough to eat, and that leads to accelerated telomere attrition. But it's also bad if you have to do a lot of stressful begging. And these 2 things are actually completely additive, which may not be clear from the graph, but if you think about it, these guys had had no bad things, and they're the highest. These guys had 2 bad things, and they're the lowest. And the guys who had 1 bad thing and not the other are about the same as each other, and they're intermediate. So again, this is a sort of, we were really pleased with this result that shows that you can actually change the cellular biology of these birds very quickly, just by 11 days of kind of systematic manipulation of the kind of parental investment that they get. Okay, and the cool thing about that is that there are actually some studies in humans that suggest really similar things. Now, measuring telomeres is more difficult in humans for a number of reasons, or from blood at least is more difficult. In birds, the red blood cells are nucleated, so we use red blood cells. In humans, you have to use white blood cells, and that causes some challenges. But for example, this is a recent paper from Idan Shalev's group. He looked at some kids at age 5 and then again at age 10, so it's like us, it's a longitudinal study. And he stratifies here by, were these kids exposed to no violence at all, 1 exposure to violence, and he's got a way of characterizing what that is, and 2 or more exposure to violence. And what you see is that the kids that are exposed to violence have a greater shortening over that period of 5 to 10. So what we're able to do in days, he can do in a correlational way over the course of years in humans, which is a nice kind of idea that the starling is a sort of speeded up model of human ontogeny. Okay, I need to press on. So the second part of my talk, so far I've been concentrating on this side of the diagram. Early life adversity, damages your adult state. But what about this side of the diagram? Your behavioral phenotype might get kind of adjusted to the kind of soma that you've got. And here I'm talking about this idea of reactive calibration. You adopt a behavioral phenotype appropriate to the kind of body you've been able to develop. And in particular, the trait I want to think about here is impulsivity, right, which has various definitions, but we can think of it as the kind of the preference for easy to get immediate rewards over harder to get more deferred ones. Might that go with having a worse somatic state as a result of your development? Now, there's evidence that it might. So in humans, for example, just growing up in a deprived postcode is associated with greater impulsivity. In a recent study that we found, this is a thing called the stop signal reaction time task. It's a very simple lab measure of a kind of impulsivity. Impulsivity has various definitions, but this is a kind of impulsivity. And basically, in this study, we just took the deprivation score of the postcode the person grew up in, so just a simpler thing as that. And these people, this was a community sample who are now in their 20s and 30s. We just took the deprivation of the postcode they'd grown up in, and we get a significant relationship between their impulsivity score, and here higher is more impulsive, and the deprivation score of the postcode they grew up in. So it's quite plausible that something about an adverse start in life could affect your impulsivity. But how do we measure it in the birds? Well, here we use something called the adjusting delay impulsivity task. It's all based on the idea that with each second that passes, the size of a reward, the subjective value of a reward is devalued, but it might be devalued at different rates for more and less impulsive people. So we keep our birds in Skinner boxes. Well, they're adapted Skinner boxes. They can fly around in them, but they have an operant panel at the end. And we train them on what we call force trials, that a green light, if they peck it, is after a delay of 1 second going to deliver them with a pellet. A red light, after a delay of X seconds, is going to deliver them a larger number of pellets. Okay? So we train them in what we call force trials. They only get one of these choices, right? So for most of the trials, they just see one, and that's so that we're sure they understand what the value of the two rewards is. But then we throw in amongst these choice trials where we actually give them both lights, but they can only peck one, and then we see which one they choose. And what we do is we use a titration procedure where if they're choosing the larger later reward consistently, we increase the length of the long delay, obviously making it kind of less attractive. And that means that the long delay goes up until eventually there reaches a point where they're not prepared to wait that long. So they start choosing the shorter, the smaller, sooner reward. And we go on this way. If they consistently choose the smaller, sooner reward, we then reduce the length of the adjustable delay, the X seconds here. So what we're trying to do is titrate to the point where the bird is indifferent, okay, between 5 pellets after X seconds is the same subjectively to it as 1 pellet after 1 second. And that allows us to calculate each individual's impulsivity. Just a little video, if we can have the video, please, of how this task works. So here's a bird in his cage. They spend all morning foraging at this task. So first of all, we're going to see the short delay forced trial. So we're just going to get one thing to choose from. So here he is. They get an initiation light to say there's a trial ready. They come down and forage at it. Green, thanks very much. I don't have to wait too long. And he gets his pellet. And then he'll have a couple of minutes off, until the yellow light comes on again and he can do more foraging. So now we're going to see the adjusting delay forced trial. So here, he presses the initiation light, he's got red. Red, he has to wait longer. It's kind of frustrating after a while. So he's learning the value of what the current value of the long delay is, right? Which is too long for this guy, as far as I can see. Eventually, thanks very much, I get my larger reward. And then interspersed with this, we give him the choice trials, right? Which you'll see now. Same deal. Yellow light comes on. I can come down and forage. Which one I'm going to choose? Oh, I think I'll have the green, thanks. And relatively soon, he gets his pellet. Okay, so what happens in this? Well, we run these birds for hundreds of trials. We start with the long delay very short, and so they favor it, and so it goes up a lot, but then it gets to a point where they can't wait that long, they can't bear it, so they switch to the shorter one. It then collapses, and you get this kind of oscillation. But we can draw a sort of mean line through these oscillatory curves, And so, for example, cage 5 here was a guy who's really not very impulsive. His long delay never gets more than about 5 seconds. But cage 1 here, he gets up to 20, 25 seconds sometimes. He's much more prepared to delay. And we can use these many trials of data to calculate something called K, which is the discounting parameter in a model of hyperbolic discounting based on where the indifference point is. And actually, we find that our K values are very different for different birds. So these are just some birds of ours by genetic family. So there's some familial effect on impulsivity. About 15% of the variance is usually explained by which genetic family they came from. But as you can see, there's also a huge amount of variation within families. So this guy has a high K, which means he discounts the future very steeply. This guy down here, in fact, this whole family are kind of somewhat patient. So generally, we get K values in the range of what is known for pigeons. A few of our starlings are as patient as rats, which is quite amazing. So the question we wanted to ask in a study I'm going to tell you about now is, does the extent to which your telomeres shortened during your early development as a measure of your kind of somatic state predict how impulsive you'll be? And our prediction is that if your telomeres shortened a lot, you're in kind of a bad way, you're going to have a preference for foraging in shorter, sooner, more easily available kind of rewards. So they're going to have a higher K value, more steep more steep discounting. So this experiment was done in a different cohort of birds from the experiments I've told you about so far. We knew from this cohort that the position in the brood when they were growing up, so these were parent-reared birds, affected their developmental telomere attrition. So the ones that were at the top of the heap, they were the biggest one in the brood, lost less telomere than the ones who were at the bottom of the heap. They were the smallest in the brood. So we already kind of knew about that relationship. And so in keeping with my general thinking about this topic, we want to know, did the extent to which your telomeres shortened, does it now predict your impulsivity? When the birds are about a year old, so just remember this, that the telomere attrition is measured over the first couple of months of life, so when they're really young. But now we're coming back, much longer than that. The birds are about a year old when they're doing the experiment. Now, what we found, which I think these are really lovely results actually, is that there were two additive influences on these birds' impulsivity, their K values. So their current body condition affected their K value in a way that I'll show you, but also the rate of telomere loss in the first couple of months of their life also predicted their K values. So here's the first of those effects. This is a measure of body condition. It's kind of like sort of BMI for birds. Higher value equals you're fatter for your skeletal size. And here's your K value. And what you see is that birds that are skinny are super impulsive, or relatively impulsive. Birds that have got good energy reserves are prepared to wait longer, have these more difficult to get but more deferred, larger rewards. And that actually plugs into a whole tradition of theorizing in behavioral ecology about how foraging should depend on your current energy budget. So that was very nice. But in addition to that, and additively, we also see that birds that had lost more telomere early in life, so that's a negative going number here, were also more impulsive. And this, as I say, was an additive relationship, which I think is really nice, because it says if we want to understand how impulsive an individual is going to be, we need to know about what's going on right now. We need to know about their current state, like in the case of a small bird, its energy reserves. But we also need to know about its long-term developmental history. Did its development just leave it in a bad way, in a more structural kind of long-term sense? So the fact that we were able to find these two effects in this study was very, very gratifying, I think. Okay, so I've given you a bit of a whistle-stop tour of some of the things that we're doing with the birds. Oh, by the way, I just wanted to add, and the nice thing about this is that a group of behavioral economists have just done a big study in humans correlating delay discounting. So, would you prefer $20 next week over $10 today, with telomere length? And they find a very similar result to ours, that people with short telomeres are more impulsive. They want the smaller, sooner amount. They discount the future more. But interestingly, their interpretation of it is completely the other way around to us, and that's maybe because they don't really have an overtly evolutionary perspective. What they say is, being impulsive is really bad, and the proof is that the people who are impulsive end up with short telomeres. Now, I think we would view this as just the other way around, right? And in fact, because we measure telomere length early in life and impulsivity later, we can rule out that direction of causality, right? We say that, if your environment has screwed you up, then you adopt a compensatory strategy of, seizing the day. So that's a kind of nice result, and we sent them our paper, and they just thought we were weird. Both for the quality of our reasoning, and also we were doing some crazy thing with 31 birds, and they just we don't know what that is. But anyway, so I've tried to give you a whistle-stop tour of what we can do with this system. We can really get experimentally at what happens to you early in life, what it's doing at the sort of cellular level, and then relate that to the behavioral phenotype that you adopt. The first thing I'd like to do is kind of advocate this way of thinking. It's not the only way of thinking, and there may be other ways of thinking about the effects of early life that have merit. I don't want to dismiss that. But I sort of like the idea that measures of somatic state, like telomere length, there might be other ones like DNA methylation, might serve as biomarkers of everything that ontogeny has done to you. And what I like about them is that they integrate different kinds of adversity, right? So psychosocial adversity, nutritional inadequacy, infectious disease, perinatal complications. These are all different kinds of adversities. But to the extent that they cause somatic damage, they might actually end up having very similar associations with behavioral phenotype, because they're just different ways that this kind of somatic state thing can come about. So I really think that studies of early life adversity could do with finding biomarkers of the kind of young adult soma, because they might actually be better than the kind of things people use like father absence and social class and so on, which seem to me, these are kind of weak indicators of something that's happening to the individual. Maybe with biomarkers we can get kind of slightly closer. So I would, I'd like to kind of advocate this way of thinking about the sort of causality. This part is pathological, and this part is adaptive. And it actually plugs nicely into a long tradition of theorizing in behavioral ecology, which says that decisions should be state-dependent, right? The behavioral strategy you adopt is not the same for all individuals. It's going to depend on short-term things like your current resources, your current energy reserves, but it's also going to depend on long-term aspects of your somatic capacity, which might be picked up by these things like telomere length or telomere attrition. The other thing I'd like to say is I'd like to sort of make a plea for more comparative work in this area. I mean, I'm sort of Johnny-come-lately to this. I've worked for 20 years on humans and only for 4 years on birds. But I personally find it exquisite, the things we can do experimentally in the avian system. That, I've written so many discussion sections to papers over my career, which are kind of disappointing, saying, we found all these associations, we're super excited, but we have to admit that we can't say anything about causality. And many of you in this room have had that experience, I'm sure. But actually, with some of these manipulations, you really can to say a bit about causality, particularly when you use these tightly controlled things like assigning 4 siblings to different developmental treatments. In most of biology, when you say to people, I'm going to take an evolutionary perspective, what they take that to mean is, I'm going to think in fitness terms. I'm going to think about the implications of phenotypes for survival and reproduction. But I'm also going to think in comparative terms. I'm going to look at how it works out in different animals. And in this community, I think we do the former, but we very rarely do the latter. So I really want to sort of stress, for me anyway, starting to do this comparative work has been kind of very exciting and allows possibilities that I hadn't dreamt of before. Just finally, we've looked at telomeres, but there are many other things you can relate to early life adversity, and we can do them all in the starling. We can look at inflammation, because there are highly conserved inflammatory markers that are the same in birds as humans. We can look at DNA methylation, which is emerging as an important biomarker of biological aging. We can look at the stress system, which is very highly conserved across vertebrates. We can look at loads of cool behavioral stuff, like cognitive biases, which are sort of measures of mood. We can look at mass regulation. We can look at risk. The whole paradigm of testing risk proneness is very well worked out in the starling. So actually, there are a whole suite of things that in the human literature have been associated with early life adversity, and we can actually look at almost all of them in the bird literature. So I'd just like to finish with these 2 characters, Wolfgang Amadeus Mozart and his starling. You may not know that Mozart had a starling that he was absolutely devoted to. And there was a very productive relationship between these two species. There are passages in Mozart's work that have been attributed to the starling's song, and the starling would also sing in particular the air from one of Mozart's piano concertos that would drive Mozart completely nuts by singing a G sharp where he wrote a G natural. But that's what the story goes. Anyway, when the starling died, Mozart gave it an incredibly elaborate funeral. He was absolutely devoted to this bird. But anyway, like Mozart, I've become convinced that we can learn a lot by having these remarkable animals, which, by the way, you only have to go outside this theatre to see. They've absolutely taken over Missouri. by going back and forth from one of these long-lived vertebrates to another, we can learn both about the biology of each, but lessons for the biology of the other one. And on that, I'd like to thank my wife, Melissa, who is the brains of the operation, and our various collaborators, both on the molecular side, on the bird side, and on my human work. And thank you for your attention.